I know what you were all thinking. A lot going on in that film, but please, I want to know more about the fish. I purposely haven't watched that film recently. I saw it probably when all of you saw it, which was about 30 years ago. And I thought that while sitting in the audience, I was going to get some glimmer of an introduction to my talk from it. I got nothing except there was a really messed up fish tank he had. There were South American cichlids in there. There was a fish that's electric in there. There's a lot going on in that tank that just wasn't right. Hold on. I bet that the electric fish would have killed him, but it wasn't, oddly, it wasn't in there when he was pulling fish out of the tank. Hold on, let's see if I can. Oh, God. Wait. Oh, I turn it on. Oh, it vibrated. Oh, boy. Help. Oh, wait. Oh. I pushed every button I got. Oh, what did you push?
I just reset it.
Oh, yeah.
I think you threw it into a mode, so it's forward.
That one, thank you.
Yep.
I've broken the theater. Okay, I want to tell you a little bit. When we were trying to come up with what I was going to talk about for this, we had a lot of choices. I work with fish, and that's what you're all going to learn about today. But I work in a rather unusual laboratory at St. Joseph's University. It's my dream lab. It's what I always wanted to do when I was about 5 years old, and somehow I figured out how to do it. And it's a place I call the Biodiversity Laboratory because we have hundreds of rare and exotic species in there. When I think about what I call normal science, at least for biologists, that would be a laboratory where you're asking one type of question and you've got one type of model organism, like a white rat in a cage or a zebrafish or nematodes or something like that. We go about science differently in my lab. We are more interested in the species than the specific questions that we're asking about them. We come up with the questions afterwards, but we want to look at lots and lots of different animals. So my laboratory looks different than most labs. It's actually 5 different spaces and a greenhouse, and it is filled with tanks, and we have lots and lots of unusual animals. We've got frogs, turtles, fish, of course, flies, some venomous snakes. This is actually one of the rarest animals in it's actually from South America, but it's very rare to find anywhere in North America. And in terms of research, we work in 4 major groups. We work with insects, amphibians, reptiles, and of course fish. So when Travis and I were talking about the possibility of me speaking here tonight, we had some possibilities. So for instance, I work with Drosophila. This is a model organism. It's what most people call a fruit fly. Later on, if you want to know, I'll tell you why it's not a fruit fly. We study sexual behavior, which is diagrammed up here. They actually have a very, very remarkably complex love life. We look at sleep, and recently we're looking at that little guy there, which is a very nasty invasive species in this area. This could have been the topic tonight, and as Travis suggested, we could have seen the fly. This was the original fly. The newer fly scares me, so I said no to that. I also work with turtles, which are my favorite animal in the world. We work with the IUCN, which is the group that determines what species are endangered and what level of endangered they are. And we run what are called assurance colonies, which are for species that are so rare they're in imminent peril of going extinct, and we are assuring that they are around by keeping them at least alive and breeding in some form, and someday maybe to be released into the wild. We could talk about that, but you would have had to watch Ninja. I've never seen this. I can't bring myself to. I also work with frogs, and we've been doing this for about 25 years. We've been studying climate change in Central America with some really remarkable frogs, which are sort of like canaries in a mine shaft in terms of climate change. They're showing the effects of it much more quickly than other things. And of course, if we'd talked about that, we would have been able to sit through the Muppet movie. And there have been some other possibilities as well. But instead, we decided to talk about fish. And I realized more so than ever watching the movie that the only thing my research has in common with this film is that the word fish is in the title. But we study social behavior in fish, which is actually rather fascinating. They're much more interesting socially than you might imagine. Fish love to be in groups with other fish, and what we study is the way they determine which groups to be with. Now, I've written a lot of papers on social behavior, and to do that, I found myself looking up the interesting names for social groups of animals. There's hundreds of them, so some of them you probably know. A group of birds is called a flock. Group of wolves, now it gets harder. Swarm, excellent. There's other names for it too, but yeah, that's good. Turtles, a group of turtles. People don't usually think of groups of turtles. It's a bale. And frogs don't usually get into groups, but a group of frogs are an army of frogs. They're also called a knot of frogs, K-N-O-T. But then we get to what's most interesting for us tonight is I want to know, what do you call a group of fish? Thank you, by the way. Wrong. And the reason I ask the question is because I want to say that. Every time I give this talk, I say something like that. Yes, indeed. That's what most people say. Actually, school is sometimes correct, but not always correct. A group of fish is actually known as a shoal. And in one old piece of writing that I found, it's thought that the word school actually arose from a misinterpretation of the word shoal. But the way we think about it today is that any group of fish is a shoal. A school is a subset. A school is a very special type of shoal. And in a school, fish swim all together in the same direction and turn simultaneously. It's something that most fish cannot do. The fish in that picture can. Fish like tuna can do it. Sardines and anchovies can do it. But most fish can't. So anytime you see a group of fish, it's definitely a shoal. It may also be a school, but not always. Now, I can explain shoaling behavior to you, but I'm afraid I've got to get incredibly scientific to do this. I hate to do this to you because we're all having fun, but I am a scientist, so we have to get really, really serious now. So the basic idea of shoaling is this. It's dangerous to be, I know this scientific stuff, it's dangerous for a fish to be alone because there's always a bigger fish. Now, in this stupid situation I have drawn up here, you actually see a perfect scenario for a predator. All predators like to attack their prey in a very specific way. Some attack from the back, some attack from the front, some attack from the side. They all have a very, very specific way they do it. Fish like to attack other fish primarily from the back, but no matter what, this situation for the predator is perfect because he's bigger than the prey, he's faster than the prey, he's going to win in a fight, but most importantly to us, he can see the prey completely, all aspects of it. So the predator controls when and how he'll attack. Oh God. There's too many buttons on this thing. Oops, I'm going backwards, hold on. I'm actually smarter than I seem. Oh, so the fish swim away. Now, let's think about shoaling behavior. That is a shoal. Now, the predator is going to have a much more difficult time picking somebody out in that. It's much safer for them to be in a group. As a matter of fact, when a predator sees something like that, if they're approaching it, they often back up. Predators do this all the time. Even sharks, when they're confronted with a huge shoal of fish, they often back away from it. They may eventually start killing things, but at first it slows down their attack. We don't ever know what an animal's thinking, but we think what's happening is the predator is looking to pick out one individual to kill, but it's very difficult to do that. So, as we say, it's safer to be in a group. However, What we study in my lab is how you make that choice. It's important to choose the right group. That little yellow guy's made a very poor choice. He has joined a shoal of fish that he doesn't look like, and so he stands out. And what you find is that if a predator sees a shoal like that, you can pretty much guess which one's going to get killed. He will pick out the one that stands out from the rest, and he will isolate it and attack it. If that little yellow guy joins that group, he's going to be in much better shape. Now, what's happening here are two phenomena. One is the thing we call the confusion effect, which is what you're seeing here. If all the organisms in the shoal look identical, we think they confuse the predator. They slowly attack down, and they reduce the chance that he's going to get anybody in that group. As opposed to what we saw in the slide a moment ago when the little yellow guy was in with the gray guys. That's a thing called the oddity effect. That means if you join the wrong shoal, you're likely to be picked off by the predator. And we actually, like that little gray guy has just done. So what we do in my lab is we ask fish how good they are at making the proper choice. So we do basic shoaling assays, and the first one of these we did in about 1990, and it was a great discovery for us in a very simple experiment. Animal behaviorists will all tell you, the simpler the project, the better. So we have a fish tank that's like this. There's glass walls at each end. This is just a 20-gallon aquarium, and we put glass walls at each end. The test fish is in the center. We want to see where the test fish goes. So in a basic shoaling assay, we did this originally with mollies. You put a group of fish in one side, and you ask where the test fish will go. And what the test fish does, it'll spend most of its time near the group of fish. If you test it now with what is the same species, but what we call phenotypically different, these are fish that look different, but they are actually the same species, and they're the same in every other way. But what we're asking this test fish to do is look at a shoal that's the wrong color shoal, but the other choice is nothing. What the fish will do is it'll spend its time with the shoal, even if it's not the right shoal. So the oddity effect is better than being alone. And then the final part of the assay is you give the fish the choice now. We've shown that it will go toward fish that it looks like. It'll go toward fish it doesn't look like. But when it's given the choice, it makes what we would consider the right choice. We've replicated this with dozens and dozens of fish, all different types, all different shapes, sizes, everything you can think of. It's remarkable how good they are at making what we would consider the proper choice. We've looked at factors that affect shoaling. Again, phenotype, which is what you look like. Body coloration, like we just saw, body pattern, body shape, body size. We've looked at species. Can you choose between different species? If you look like them or if you don't look like them, does that matter? Sex. In some fish, the males and females look differently. We see some interesting things there. Usually the females group together for protection. The males go where the females are, even if it's going to invoke the oddity effect. And then familiarity, it turns out that fish like to be with fish who they personally know. And we haven't figured out how they do this yet, but if you give them all the same looking fish, they can pick out their buddies and go hang out with them. So these are all factors that we see affecting shoal mate choice. The big question for us, though, is how does a fish know what it looks like? We'll never know. This is a question I don't think we can ever answer. Is there a genetic template? Is it born with its own image in it? Does it look at itself? Does it learn somehow? Or does it imprint, which is a different type of learning? What we have shown is if you raise fish in total isolation from the egg and let them grow up alone and then put them in the test, they always make what we would call the right choice. However, if you take those babies and you stick them in the wrong family with a bunch of babies that are a different color, they will then learn to go toward the other color. So it appears to be genetic unless you give them something to learn. So we're not exactly sure what's going on there. The wonderful thing about shoaling behavior is you can always come up with more questions to ask. What we're looking at right now is shoal size, and there's a thing called the numerical dilution hypothesis. It's simple. If you're in a shoal of 5, you've got a 1 in 5 chance of being killed by a predator. If you're in a group of 10, you have less of a chance of being killed by the predator. If you're in a group of 100 and so forth. So the idea is that a larger shoal, all other things considered the same, the better off you'll be. So basically we're asking if fish can count. And it turns out they can, But ratios are critical. So in this situation here, this fish would be given a choice between 2 fish of the same type or 4, 2-to-1 ratio. They always get that right. They choose the larger group. If you give them a 1.5-to-1 ratio, 2 versus 3, they never get it right. They show no inclination to choose either side. And you can work this up in any multiple you want. 8 versus 4, 10 versus 5, or in this case, 4 versus 6. Anytime you get a 2-to-1 ratio, they can do it. Anytime you don't have a 2-to-1 ratio, they can't do it. We're looking at larger groups now to see just at what point this breaks down. But at the moment, it appears that something about ratios allows them to discern between 2 groups. Now, the work we're doing this summer adds another factor to that. We're looking at mixed phenotype shoals and also looking at them in ratios. These are platies, Mexican fish. They come in a variety of colors. We've already shown that the red ones like to shoal with the red ones and the blue ones like to shoal with the blue ones. But now what we're doing is we're mixing the shoals, red and blue on each side, and we're asking if they can go to the side that has more members of their own phenotype. And as you might have guessed, they can do it in a 2-to-1 ratio. So 4 versus 2, they can do it. 3 versus 2, they cannot do it. And we're continuing this summer. We're going to look at a whole series of other things having to do with that as well. So the conclusions of my quick little talk on shoaling. Shoaling confers a number of benefits. Some we didn't talk about, but if you're in a big group of fish, you find food more quickly. You could have the potential of finding mates. And then, in terms of predation risk, size matters, the shoal anyway, and you must choose your shoal mates wisely. And Wanda needed some more angelfish in the tank, maybe for some protection. Anyway, thank you very much. This has been very nice coming and talking to you. And if anyone has any questions on this, Kevin Klein, frogs, whatever. Yeah.
So since shoaling provides so much protection, why don't most fish necessarily, swim in shoal? There are several types that do not, right?
There are. The question is, why don't most fish join shoals? It turns out that from what we're looking at, most fish that are under high predation pressure do choose shoals. Larger fish, solitary fish, are usually predatory fish, and they don't have the reason for it. The other thing that is a little bit another aspect to this, especially we see in coral reefs. Fish often in coral reefs are dispersed around the reef, and if you take photographs of a coral reef at any one time, you'll see fish all over the place. If you scare them, if you bring a predator in, or if you do something to frighten them, they suddenly group together. So sometimes fish swim in a very loose organization, but under threat, they come together. And we've actually done that in some of our test tanks. We've done that test we have, but behind the center panel, we had a screen that we could pull up with a predator in the fish tank, and it's amazing how quickly they will rush to one side. Anything else? Yes? I haven't looked at humans. My training is in genetics, and the last thing they do before they let you out the door getting a PhD in genetics is they say, make sure under no circumstances you ever compare yourself, anything you're doing, to humans. For the fish and humans, I don't know anything about humans in general. However, humans have a whole series of other capabilities. Being in groups probably is a tremendous value, but for a very different reason. We're not trying to blend in from our predators, who are often ourselves. Grouping in humans, I think, confers a ton of benefits, but probably in a very different way. There are lots of other species of animals that do group like this. Birds do it. There's some insects that do it. There's a lot of mammals that do pull themselves into tight groups. A lot of arboreal primate species pull themselves into groups under threat. But I don't know of anything quite like this with people. I think it's different with us. Yeah. Oh, thank you. How sweet. Thank you so much. Okay, thank you. Thank you so much. Yeah. Excuse me? Music. The fish? Well, wait a second. I was looking at my platies the other day, and I had Jimi Hendrix blaring, but they didn't seem like it mattered. I do not know. I've never done anything with sound specifically in fish, and whether it's we've done a lot of things with light, but I don't know. Yeah. Yeah. Yes. That's actually a really interesting thought. In those tanks we have, they can't get together. There's a glass wall, and the test fish swims back and forth in the center. What we're doing is spending the amount of time it spends in close proximity to either side. But what you find is when the test fish goes up to the glass, it will put its mouth along the glass and go up the glass. The fish in the shoal will often come over and they attend each other. It's like they're trying to touch each other. So the ones inside are responding to the single individual as well. I think they're trying to form a group, but then the glass prevents it. The question is whether it's the same if the one in the center is different. You know what? I actually don't know. It's just anecdotal that I've watched them do that, but I actually don't know. That's an interesting question. The wonderful thing about fish, my students will say this all the time. Every time you talk about this, you can come up with a new idea. It's my type of science. It's very simple. You can just automatically come up with a new idea. Someone was saying in a seminar I gave last week, why don't you put two fish in the middle? And my response was, well, statistically, that's a nightmare. But then as soon as I got that stupid sentence out of my mouth, I thought, boy, that would be really cool. So the heck with the statistics. But, I think I'll have my students take a peek at that this summer. They're doing the platy study, and there's going to be a lot of opportunities of a red platy up against a group of blue and vice versa. I'll have to take a look and see if they see anything just anecdotally about the response to the group inside. If there's something there, that's a new study. Yeah. Very much, I think they probably are. I have a feeling that when they get together with a member coming into a new group, they probably touch, and there's probably a lot of communication. And I think that when one comes up to the glass, as soon as that happens, they're prevented from doing something. They're very attentive to the group on the other side. So I think absolutely they're communicating information with each other. Yeah.
Is there ever an upper limit on the shoal size where the fish maybe is worried about lack of resources or avoids a group that's too big?
That's actually a great idea. The question is whether you join a group that's too large, whether there'll be diminishing returns because now you're in more competition. I don't know anything in the literature that talks about that. And anybody who does tests like the ones I do, we're always working in a really rarefied scenario. It's a laboratory setting, and we're always looking at small fish. We've only just recently decided to try to do that numerical thing with much bigger numbers. But initially, my thought for that was, if you were looking at 50 fish versus 25, does it get to a point where they can't discern the difference? But the idea of whether you would present fish with groups that were so large that maybe joining the big group has a different parameter to it, I don't think anyone's ever approached that. You want to work in my lab? That's actually, seriously, that's actually an interesting thought, because you think that the basic natural selection phenomena is every time groups get large enough, they fracture in order to find ways of allocating resources properly. I don't think anyone asked that with fish. It's good stuff. Yeah. It is. This is a good group. I agree. Very good question. Actually, I think there's going to be some papers come out of this at some point. It's very, very good. Yeah. I'm sorry. No, they're all shoaling. We never do anything with predators anyway. So if you had the wrong type of fish on the other side and you remove the glass, yeah, it would be bad. These guys are, we never work with animals that would view each other in that way. So for our fish, if they got together, they wouldn't. Now, we did a series of experiments on Siamese fighting fish. You know, bettas, they sell them in the pet shops. That's a very unusual fish. The males are highly aggressive toward images of themselves. They'll fight with mirrors, they'll fight with other males. What most people don't know is the female bettas are very interesting animals. They rarely sell them in pet shops. They're not nearly as colorful as the males. Females are also incredibly aggressive, but they form shoals. And so we did a series of tests where we asked the males, would you like to be with the girls or would you like to fight? And we found out that if you sized the males and the one in the middle was bigger than the male on one side, he'd rather go fight. And if you put him in the tank and the guy on one side was bigger than him, he'd rather go be with the girls. And they seemed to size that up over about 5 minutes of back and forth in the tank, and then they would just stay at one side or not. And the female bettas are the only fish we've ever seen that does the converse to shoaling. If you give them other females, they want to do anything to stay away from them because they're very aggressive to each other in groups. But for our fish, no predators. If there was, that would be bad. Oh, yep. It's hard to see out there, sorry. Yeah, so it's a good question. So the idea is, I'll have to say it the way you said it, if you give it a choice between a different species, but it's the same phenotype, or the same species and a different phenotype, it will choose to be with the fish that it looks like. So, and you can do that with a whole series of these platies and things. The color or the body shape or the pattern is more important than species. The only time species kicks in is if you're looking at some slight differences, or if everyone looks the same or very similar, then they can pick out species. But phenotype overrides species. Of course. Mm-hmm.
That butterfly fish will understand mate for life, and that they can differentiate between and recognize a person. Is there any evidence that this is possible?
Recognize a person. I wouldn't discount that idea at all. So we don't do anything like, we do recognition of individuals fish to fish, and it does appear that if you house, like, you take a bunch of zebrafish that live together for a few months, you take one of them out, put it in the test tank, you give it 3 zebrafish, they all look exactly alike, that it doesn't know, and 3 that it does know. It goes to the ones it knows. We don't know how it does that, and to be honest, it's a quirky experiment, doesn't always work. As far as recognition of humans, that has been shown in a lot of different species, and we've actually worked on it in my laboratory with lizards. We had a great big iguana named Fido who's found himself in a number of different publications. He was able to pick out people based on their face. It led to, it was me. It was an inadvertent study. he bobbed his head every time I came in the room, which is actually a threat display, and he would ignore everyone else. So we concocted this remarkable experiment. We needed to do something and then count the number of times he bobbed his head. He was in a huge cage at the back of the lab. And so we decided we wanted to send people in one at a time, and they would do something to interact with the lizard, and we'd see if he bobbed his head. So we read him the book, Oh, the Places You'll Go, Dr. Seuss book. And so the book, and we had everybody in the department get involved with this. And so for an experiment, you'd walk into the lab, sit down in the chair, pick up this book, and begin reading. And there were cues in the book that every now and then you would do something which is considered somewhat threatening to a lizard. Fido'd just be sitting up on his perch. You'd stand up and lean toward him in a menacing way, and then sit back down. Everybody in the department who did it, he would just sit there and fall asleep. And if I did it, he'd bob his head like 300 or 400 times. We think it's because I went into his cage all the time to clean it and feed him. I was actually taking care of him, but I was in his territory. So then my students came up with 2 cockamamie studies that we did. One was they took a picture of me and they made masks out of it. So people, it was disconcerting. I'd come to work and see people walking down the hall with my face. And then they did, and that never worked. It had to be me. And then they did a thing where we lip synced. We had people read the book, and then you lip sync to it. It still had to be me in person. Then one of my students decided that it was because I was the boss of the lab, and that Fido, being a dominant male, knew I was the guy to take out. So for about 2 weeks, we did this experiment that one of my students absolutely loved, where he berated me mercilessly in front of the lizard. He'd yell at me. He'd make me pick things up off the floor. He'd spill things intentionally and make me mop them up. So we thought, we'll convince Fido that I have been displaced by this person. But it didn't work. But stupid answer to a good question. There is evidence also in the wild for this. There's a series of iguanas that live at the airport in San Juan, Puerto Rico, and people feed them, and the lizards have learned to run across the tarmac to people that they know have fed them in the past. So they do that. So I'm sure a fish could do that as well. Yeah. Good. Thank you so. Oh wait, another one, or just thank you so much. This has been really, really great fun. Thank you.